Gene editing, mechanisms, technologies, and approaches. The CRISPR, clustered regularly interspaced short palindromic repeats, system has been a groundbreaking tool in genetic engineering, offering precise modification to DNA that can lead to advancements in medicine, agriculture, and more. However, there are other systems and technologies developed or being developed for gene editing and genetic manipulation, each with its own advantages and limitations. Zinc finger nucleases, ZFNs. Zinc finger nucleases, ZFNs. ZFNs are proteins engineered to target specific DNA sequences. They work by fusing zinc finger DNA binding domains with a DNA cleavage domain. ZFNs can be designed to target specific genomes, allowing for precise gene editing. However, designing and constructing ZFNs can be complex and costly. Zinc finger nucleases, ZFNs, are powerful tools for genome editing that operate through a combination of DNA binding and DNA cleavage mechanisms. Their function involves several key biochemical steps: zinc finger protein, ZFP, DNA binding. ZFNs consist of a fusion between a zinc finger protein, ZFP domain, and a DNA cleavage domain. The ZFP domain is engineered to specifically recognize and bind to a particular DNA sequence. This specificity is achieved through the modular nature of zinc finger motifs, each typically recognizing 3 base pairs of DNA. Zinc fingers are small protein motifs that stabilize with the help of a zinc ion. Each motif can be designed or selected to bind to specific triplets of DNA bases, allowing for a customizable recognition sequence by stringing together several zinc fingers in a single protein. Foci nuclease domain, dimerization and cleavage. The DNA cleavage domain is derived from the foci restriction enzyme, which requires dimerization to become active. Therefore, ZFNs work in pairs, with each ZFN binding to its target sequence, and the foci domains coming together to form an active dimer. Once dimerized, the foci nuclease domains introduce a double-strand break, SB, in the DNA at a site between the binding sites of the paired ZFNs. This break is typically a few base pairs away from the ZFP recognition site. DNA repair mechanisms. The cell repairs the DSB through either non-homologous end joining, NHEJ, or homology-directed repair, HDR. NHEJ can lead to insertions or deletions, indels, at the repair site, which can disrupt or modify the function of a gene. HDR, in contrast, can introduce specific mutations or insertions by using a donor DNA template with homology to the sequences flanking the DSB, allowing for precise genome editing. Specificity and off-target effects. The specificity of ZFNs depends on the design of the zinc finger motifs and the recognition sequence. Mismatches between the ZFN recognition site and the target DNA can affect binding affinity and specificity, potentially leading to off-target effects where the ZFNs bind and cleave unintended sites in the genome. Optimization for genome editing. Extensive research has been conducted to optimize ZFNs for higher specificity and efficiency in genome editing. This includes engineering ZFPs for more specific DNA binding, optimizing foci cleavage domain variants to require heterodimerization, reducing off-target cleavage by homodimers, and improving delivery methods to target cells. ZFNs have been used in various applications, including gene therapy, functional genomics, and the generation of genetically modified organisms. However, despite their utility, the complexity of designing specific zinc finger arrays for new target sequences has led to the development of other genome editing tools like TALENs and CRISPR-Cas systems, which offer simpler design and comparable or improved specificity and efficiency. TALENs, transcription activator, like effector nucleases. TALENs, transcription activator like effector nucleases. Similar to ZFNs, TALENs use customizable proteins for DNA binding, paired with a nuclease for cutting DNA at specific sites. TALENs offer more flexibility and simplicity in targeting specific DNA sequences compared to ZFNs, but still require complex design and engineering. Transcription activator like effector nucleases, TALENs, are a class of engineered enzymes used for genome editing, similar to zinc finger nucleases, ZFNs, but with a different mechanism for DNA binding. TALENs operate through a series of well-characterized biochemical mechanisms, DNA binding through tail domains. The DNA binding domain of TALENs is derived from transcription activator-like effectors, tails, found in Xanthomonas bacteria, which infect plants. Tails bind to specific DNA sequences via a central region of tandem repeats, each consisting of approximately 35 amino acids. Each repeat is highly similar except for two variable amino acids, known as the repeat variable diresidue, RVD, which determine the DNA base that the repeat will bind to. This one-to-one correspondence between the RVD and DNA bases allows for precise targeting of specific DNA sequences by engineering tail repeats to match a desired sequence. Foci nuclease domain. Cleavage. Similar to ZFNs, TALENs utilize the Foci nuclease domain for DNA cleavage. However, the foci domain is inactive as a monomer and requires dimerization to become active. Thus, TALENs are designed in pairs to bind to opposite strands of DNA, bringing two foci domains into close proximity to dimerize and introduce a double-strand break, DSB, in the targeted DNA site. The DSB occurs within the spacer region between the TALEN binding sites on the DNA. DNA repair mechanisms. The introduction of a DSB by TALENs activates cellular DNA repair mechanisms. The cell may use non-homologous end joining, NHEJ, to ligate the break, often resulting in insertions or deletions, indels, that can disrupt gene function. Alternatively, if a donor DNA template is provided, the cell can use homology-directed repair, HDR, to introduce specific changes or insertions at the break site, allowing for precise genome editing. Design flexibility and specificity. The modular nature of tail repeats and the clear relationship between RVDs and DNA bases make TALENs highly customizable and specific for targeting almost any DNA sequence. This has made TALENs a powerful tool for genome editing across a wide range of organisms and applications. The specificity of TALENs can be further enhanced by designing longer tail arrays or using variants of the foci domain that require heterodimerization, minimizing off-target cleavage by reducing the activity of foci homodimers. Applications and limitations, TALENs, have been used in a variety of applications including gene knockout, gene insertion, and correction of genetic mutations. They are particularly useful in cases where high specificity is required or when targeting sequences with less flexibility for PAM sequences, as required by CRISPR-Cas9 systems. While TALENs offer high specificity and versatility, their design and assembly can be more technically challenging and time-consuming than some other genome editing technologies, such as CRISPR-Cas9. The size of TALEN constructs can also pose challenges for certain delivery methods. Overall, TALENs represent a powerful and versatile technology for genome editing, with their mechanism of action allowing for precise modifications of genomic DNA. Their development has significantly advanced the field of genetic engineering, contributing to research in genetics, developmental biology, disease models, and potential therapeutic applications. Meganucleases. Meganucleases, also known as homing endonucleases, these are enzymes that recognize long DNA sequences, providing high specificity for targeting. Meganucleases can be engineered to cut specific DNA sequences, facilitating gene editing. Their limitation lies in the difficulty of engineering them to target new sequences. Meganucleases, also known as homing endonucleases, are a family of highly specific DNA-cutting enzymes that have garnered interest for their potential in genome editing applications. These enzymes recognize long DNA sequences, 14 to 40 base pairs, which allows for a high degree of specificity in targeting genomic DNA. Here are the known chemical and biochemical mechanisms by which meganucleases work. DNA recognition and binding. Meganucleases contain several DNA binding motifs that allow them to recognize and bind to specific DNA sequences. These sequences are longer than those recognized by other DNA-binding proteins, contributing to the high specificity of meganucleases for their target sites. The binding mechanism involves multiple contacts between the enzyme and the specific bases within the DNA sequence, as well as interactions with the DNA backbone. This extensive network of interactions stabilizes the enzyme DNA complex and positions the enzyme for cleavage. DNA cleavage. Upon binding to their target DNA sequence, meganucleases induce a break in the DNA strand. This is achieved through the catalytic activity of the enzyme, which typically involves the coordination of a metal ion, such as Mg2+ or K2+, in the active site. The metal ion aids in the hydrolysis of the phosphodester bond, resulting in a double-strand break, DSB. The cleavage mechanism often produces blunt ends or overhangs of a few nucleotides, depending on the specific meganuclease and its mode of action. DNA repair and genome editing. The introduction of a DSB by a meganuclease triggers the cell's natural DNA repair mechanisms. Cells can repair the break using non-homologous end joining, NHEJ, which can introduce insertions or deletions at the site of repair, potentially disrupting or altering gene function. Alternatively, if a DNA template with homology to the sequences flanking the DSB is provided, the cell can use homology-directed repair, HDR, to introduce specific sequence changes or insertions, enabling precise genome editing. Engineering and customization. Natural meganucleases can be engineered to recognize different DNA sequences, enhancing their versatility for genome editing applications. This is achieved through protein engineering techniques that modify the DNA binding domains of the enzyme to alter its specificity. The customization of meganucleases, however, is technically challenging due to the complex interactions between the enzyme and its DNA target. Applications in gene therapy and biotechnology. Due to their high specificity and efficiency, meganucleases have been explored for various applications in gene therapy, including the correction of genetic mutations and the generation of genetically modified organisms. Their ability to induce targeted DSBs with minimal off-target effects makes them valuable tools for precise genome modifications. Meganucleases represent an important class of enzymes in the field of genome editing, offering high specificity and the potential for therapeutic applications. Despite the challenges in customizing these enzymes for new target sites, ongoing research and development continue to expand their utility in genetic engineering and biotechnology. Base editing. Base editing. Developed as an alternative to the standard CRISPR-Cas9 system, base editors directly convert one DNA base into another without creating double-stranded breaks, potentially reducing the risk of unwanted mutations. Base editing allows for more precise genetic modifications at the single nucleotide level. Base editing is a powerful genome editing technology developed as an alternative and complement to the traditional CRISPR-Cas9 system. It allows for the direct, irreversible conversion of one DNA base pair into another, without inducing double-strand breaks, DSBs, thereby reducing the risk of undesired outcomes associated with DSB repair pathways. Here are the known chemical and biochemical mechanisms by which base editing operates. Targeted DNA binding. Base editors are fusion proteins that combine a catalytically impaired CRISPR-Cas9 variant, often dCas9 or Cas9-Nicas, with a base deaminase enzyme. The CRISPR-Cas9 component is guided to the target DNA sequence by a guide RNA, a tgRNA, ensuring specificity in binding. The catalytically impaired Cas9 variants bind to the DNA without introducing DSBs. Base deamination. Once the base editor is bound to the target site, the deaminase enzyme modifies the target nucleotide. There are two primary types of base editors: cytosine base editors, CBEs, and adenine base editors, ABs. CBEs use a cytidine deaminase to convert cytosine C into uracil U, which is then processed by cellular mechanisms as thymine T during DNA replication or repair, effectively achieving a C to T or G to A on the complementary strand, edit. ABs employ an evolved adenosine deaminase that works on RNA. adenosine deaminase acting on RNA, or ADAR, to convert adenine A into inosine 1, which is treated as guanine G by the cellular machinery, resulting in an A to G or T to C on the complementary strand. Edit. DNA replication and repair. The introduction of uracil or inosine into the DNA triggers the cell's natural DNA repair pathways. Since uracil is recognized as thymine during DNA replication, the UG mismatch is resolved to a TA base pair. Similarly, inosine pairs like guanine leading to the resolution of an IC mismatch to a GC base pair. Specific DNA repair proteins, such as uracil DNA glycosylase, UDG, which is part of the base excision repair BR pathway, are involved in processing the uracil introduced by CBEs. However, the use of uracil glycosylase inhibitors, UGIs, in some CBE designs can inhibit UDG activity and improve the efficiency of C2T editing. Nicase activity. Some base editors incorporate a Cas9 nicase, NCAS9, that introduces a single-strand break NIC in the non-edited DNA strand. This NIC can stimulate the DNA repair machinery to use the edited strand as a template, increasing the efficiency of incorporating the base edit into both DNA strands. Off-target effects and editing window. The specificity of base editing depends on both the guide RNA, which determines the target site, and the activity window of the deaminase enzyme, which defines the nucleotides susceptible to editing. While base editors are designed to minimize off-target effects, the deaminase component can act on DNA within a certain range, usually 4 or 5 nucleotides, of the binding site, which may lead to unintended edits if off-target binding occurs. Strategies to improve specificity include engineering high-fidelity Cas9 variants, optimizing gRNA design, and developing deaminase variants with narrower activity windows or reduced off-target activity. Base editing represents a significant advancement in genome editing technology, offering the ability to make precise single nucleotide changes without the risks associated with creating DSBs. This technology has wide-ranging applications, from basic research to therapeutic development, enabling precise genetic modifications with potentially fewer unintended consequences. Prime editing. Prime editing. This is a newer, more precise version of CRISPR that allows for direct writing of new genetic information into a specified DNA site. It combines a Cas9 nicase with a reverse transcriptase enzyme, enabling precise edits without double-stranded breaks and with fewer byproducts. Prime editing is an advanced genome editing technology that represents a significant innovation beyond traditional CRISPR-Cas9 systems. Developed by Andrew Anzalone and colleagues in 2019, prime editing offers the capability to precisely insert, delete, and replace DNA sequences without relying on double-strand breaks, DSBs, or donor DNA templates. Here are the known chemical and biochemical mechanisms by which prime editing operates. Prime editing guide RNA, Maïwe Grenet. Prime editing uses a specially engineered RNA molecule known as the prime editing guide RNA, Grenet. The pgRNA contains both a sequence that guides the Cas9 nuclease to the target site, similar to traditional sgRNA, and a primer binding site, at PBS sequence, along with a template sequence for the desired edit. The PBS is complementary to the target site and allows the pegRNA to hybridize to the nicked strand, initiating the editing process. Nicase Cas9, NCAS9, and reverse transcriptase. Fusion protein, a fusion protein consisting of a Cas9 nicase, NCAS9, which introduces single-strand breaks instead of DSBs, and a reverse transcriptase, RT, is directed to the target DNA site by the pegRNA. The NCAS9 component of the fusion protein nicks the target DNA strand opposite the PBS, while the RT uses the 3' end of the nicked DNA as a primer to copy the template sequence from the pegRNA into the target site. Reverse transcription, After nicking, the exposed read sentence into its end of the DNA strand invades the pegRNA's PBS and hybridizes with it. The reverse transcriptase then extends the 3-malatoine end of the nicked DNA using the pegRNA's edit template, synthesizing DNA that contains the desired edit directly into the target site. Strand displacement and DNA repair. The synthesis of the new DNA strand displaces the downstream DNA, creating a flap. The cellular machinery repairs the flaps and integrates the newly synthesized DNA, incorporating the edit into the genome. This process can involve the removal of the displaced strand, containing the unedited sequence, by endogenous DNA repair enzymes, followed by ligation to complete the integration of the edited sequence. Second nicking. Some prime editing systems include an additional sgRNA that directs Cas9 to nick the non-edited strand. This second nicking stimulates DNA repair pathways to resolve the mismatch between the edited strand and the original non-edited strand, enhancing the efficiency of incorporating the edited sequence into both DNA strands. Versatility of edits. Prime editing can mediate a wide range of genetic alterations, including insertions, deletions, and all 12 possible base-to-base conversions, with high precision and fewer off-target effects compared to traditional CRISPR-Cas9 systems. This versatility is due to the direct synthesis of the edited sequence into the target DNA without the need for DSBs or donor DNA templates. Limitations and optimization. Despite its versatility, prime editing efficiency can vary depending on the target site, the specific edit, and the cell type. Ongoing research aims to optimize the components of prime editing, such as the pegRNA design, the Cas9-RT fusion protein, and the editing conditions, to enhance its efficiency, specificity, and applicability across different biological systems. Prime editing's ability to introduce precise genetic modifications without inducing DSBs marks a significant advancement in genome editing technology, opening new possibilities for research, therapeutic development, and biotechnology applications. CRISPR-Cas12 and Cas13. These are variants of the CRISPR system, discovered after Cas9. Cas12 and Cas13 have unique properties that make them suitable for different applications. For example, Cas13 targets RNA instead of DNA, opening new avenues for gene editing at the RNA level. CRISPR-Cas12 and Cas13 are two classes of CRISPR systems that offer unique mechanisms and applications in genome editing and RNA targeting, extending the versatility of CRISPR technologies beyond the well-known CRISPR-Cas9. Here are the known chemical and biochemical mechanisms by which CRISPR-Cas12 and Cas13 work. CRISPR-Cas12, previously known as CPF1, DNA targeting and cleavage. CRISPR-Cas12 recognizes and binds to a specific DNA sequence adjacent to a protospacer-adjacent motif, PM, different from that recognized by Cas9, typically a T-rich sequence. Upon DNA binding, Cas12 induces a double-strand break, DSB, within the target DNA. Unlike Cas9, which produces blunt-ended cuts, Cas12 generates staggered cuts with overhangs, which can influence the efficiency and outcomes of the DNA repair process. RUVC-like nuclease activity. Cas12 possesses a RUVC-like nuclease domain responsible for both strands cleavage. This differs from Cas9, which requires HNH and RUVC nuclease domains to cleave the complementary and non-complementary strands respectively. CRISPR RNA and CRRNA. Processing. Cas12 has an additional unique feature of processing its own RNAs from a long precursor transcript. This autocatalytic activity allows Cas12 to be used in applications requiring multiplexing, targeting multiple genes simultaneously, without the need for separate RNA processing steps. CRISPR-Cas13, previously known as C2C2. RNA targeting and cleavage. CRISPR-Cas13 is distinct in its ability to target and cleave RNA molecules, rather than DNA. This RNA targeting capability allows for applications in RNA editing, tracking, and regulation, as well as diagnostics. Cas13 recognizes its target RNA via a cRNA guide and cleaves the RNA at sites adjacent to the recognition site. The cleavage is promiscuous, meaning it can cut the target RNA at multiple sites. Collateral activity. Upon binding and cleavage of its target RNA, Cas13 exhibits a collateral or trans cleavage activity, whereby it non-specifically cleaves other RNA molecules in the vicinity. This property has been harnessed for sensitive detection of RNA molecules in diagnostic applications, such as in the Sherlock platform. HEPN nuclease domains. The RNA cleavage activity of Cas13 is mediated by two higher eukaryotes and prokaryotes nucleotide binding HEPN domains. These domains are responsible for the catalytic activity that cleaves RNA. General mechanisms for both Cas12 and Cas13. Guide RNA complex formation. Both Cas12 and Cas13 systems rely on the formation of a ribonuclear protein complex with their respective cRNAs, which guide the enzymes to their specific target sequences based on complementary base pairing. PAM, or protospacer flanking site, PIPFS. Cas12 and Cas13 systems require specific sequences adjacent to the target site. PM for Cas12 and PFS for Cas13 for efficient target recognition and binding. These motifs are essential for distinguishing self from non-self genetic material in bacteria. The distinct properties of CRISPR-Cas12 and Cas13 expand the toolkit available for genome and transcriptome engineering, offering new approaches for precise genetic modifications, multiplex gene regulation, and diagnostics. Their unique mechanisms and functionalities continue to be explored and harnessed for innovative applications in biotechnology, medicine, and research. Oligonucleotide-directed mutagenesis, ODM. Oligonucleotide-directed mutagenesis, ODM. This technique involves the use of short DNA or RNA oligonucleotides to direct specific mutations in the genome. It's a more traditional approach and is used for subtle genetic modifications, such as correcting single nucleotide polymorphisms. Oligonucleotide-directed mutagenesis, ODM, is a technique used in molecular biology to introduce specific mutations into a DNA sequence. This method relies on synthetic oligonucleotides, short, single-stranded DNA fragments designed to hybridize to a specific region of the target DNA, with the desired mutation included within the sequence. The mechanisms by which ODM works involve several key biochemical steps. Synthesis of mutagenic oligonucleotides. ODM begins with the synthesis of a mutagenic oligonucleotide, which is a short DNA fragment designed to be complementary to the target DNA sequence, except for the nucleotides where mutations are to be introduced. This oligonucleotide can carry substitutions, insertions, or deletions. Hybridization to target DNA. The mutagenic oligonucleotide is introduced to the target DNA under conditions that allow for its hybridization to the complementary strand, aligning such that the site of the desired mutation is paired with the template strand. This step often requires the DNA to be single-stranded or partially denatured to facilitate binding. DNA repair and replication. Once hybridized, cellular DNA repair and replication mechanisms are exploited to incorporate the mutation into the DNA. Depending on the method used, this may involve mismatch repair. The cell's natural mismatch repair system recognizes the mismatch between the mutagenic oligonucleotide and the target DNA as an error and corrects the native strand to match the oligonucleotide, thereby incorporating the mutation. Extension and ligation. DNA polymerases extend from the 3' to end of the oligonucleotide, and DNA ligases seal the next, with the mutation being included in the newly synthesized DNA. Strand displacement synthesis. Some methods involve the use of polymerases that can displace the downstream strand, synthesizing a new strand that includes the mutation. Selection or screening for mutants. After the mutagenic oligonucleotide has been integrated, cells or molecules containing the mutation need to be identified and isolated from those that did not incorporate the mutation. This often involves selection markers, restriction sites, or PCR amplification with specific primers. PCR-based ODM. A variation of ODM uses PCR, polymerase chain reaction, to amplify the entire gene or DNA segment containing the mutation introduced by the oligonucleotide. This approach can increase the efficiency of mutagenesis and facilitate the identification of mutated sequences. Site-directed mutagenesis. While not always classified directly under ODM, site-directed mutagenesis techniques that use oligonucleotides to introduce mutations during PCR amplification share similar principles. Here, the oligonucleotide primers carry the desired mutation, and the entire plasmid or DNA fragment is amplified to incorporate the mutation throughout. ODM is a versatile tool for genetic engineering, allowing for precise modifications at specific sites within a gene or DNA sequence. The technique has been fundamental in studying gene function, protein engineering, and the development of genetically modified organisms. Advances in oligonucleotide synthesis, DNA repair understanding, and molecular cloning have continued to refine and expand the applications of ODM in research and biotechnology. Piggyback transposon system. Piggyback transposon system. This system utilizes a cut-and-paste mechanism to insert or remove large sequences of DNA. It is highly efficient and can be used for the integration of large DNA sequences into genomes, making it useful for gene therapy and functional genomic studies. The piggyback transposon system is a versatile tool used in genetics and molecular biology for inserting genes into chromosomes. It originates from the cabbage looper moth Trichoplusia nii and has been adapted for use in a wide range of organisms, including mammals. The system relies on a cut-and-paste mechanism mediated by the piggyback transposase enzyme, which recognizes specific terminal repeat sequences in the piggyback DNA element and facilitates its integration into the host genome. Here are the known chemical and biochemical mechanisms by which the piggyback transposon system works. Recognition of terminal inverted repeats, TRs. The piggyback transposon is flanked by terminal inverted repeats, TRs, which are specific DNA sequences recognized by the piggyback transposase enzyme. These TIRs are essential for the excision of the transposon from its original location and its insertion into a new site. Transposase binding. The piggyback transposase enzyme binds to the TIRs, bringing together the ends of the transposon. This enzyme is the only protein required for the transposition process, recognizing the TIRs through its DNA binding domain. Excision of the transposon. Upon binding, the transposase catalyzes the excision of the transposon from its original location by introducing a double-strand break, DSB, at each end of the transposon. The enzyme makes precise cuts, ensuring that the entire transposon is excised without any alterations. Target site selection and integration. The piggyback system typically integrates into TTAA sites within the host genome. The transposase carries the excised transposon to a TTAA site and integrates it into the genome through a cut-and-paste mechanism. The choice of TTAA sites as target sequences is a distinctive feature of the piggyback system, contributing to its utility in genetic engineering. During integration, the transposase makes a staggered cut at the TTAA target site, creating short overhangs that facilitate the insertion of the transposon. Repair of DNA at integration site. The host cell's DNA repair machinery repairs the gaps at the integration site, stabilizing the transposon within the genome. This repair process is precise, with the TTAA sequence duplicated at the insertion site, flanking the transposon. Seamless excision. One of the unique features of the piggyback transposon system is its ability to excise from the genome without leaving behind any footprint. This is due to the precise excision mechanism mediated by the transposase, which can reverse the integration process without altering the surrounding host DNA. This feature is particularly valuable for applications where transient expression of a transgene is desired, or when generating clean gene knockouts. Applications. The piggyback transposon system is widely used in genetic studies for gene delivery, transgenesis, and functional genomics. Its ability to efficiently integrate large DNA fragments and its footprint-free excision capability make it a powerful tool for creating transgenic organisms, gene therapy vectors, and insertional mutagenesis libraries. The piggyback transposon system's high efficiency, large cargo capacity, and minimal genomic footprint have made it a preferred choice for many applications in genetic engineering and biotechnology. Its mechanisms of action leverage the natural process of transposition, adapted for precise and reversible genetic modifications. RNA interference, or RNAi. RNA interference, RNAi. Although not a gene editing tool in the traditional sense, RNAi can effectively silence gene expression. It uses short interfering RNA siRNA molecules to target and degrade specific mRNA molecules, preventing the translation of certain proteins. RNA interference, RNAi, is a biological process in which RNA molecules inhibit gene expression or translation by neutralizing targeted mRNA molecules. It's a vital method of post-transcriptional gene silencing, PTGS, that organisms use for various purposes, including defense against viruses, regulation of gene expression, and development. The RNAi pathway involves several key steps and components. dsRNA initiation. RNAi is initiated by double-stranded RNA, dsRNA, which can be introduced into cells experimentally or produced internally as part of the cell's normal processes. In some cases, the dsRNA comes from viral infections, transposons, or artificial introduction for experimental purposes. Dicer processing. Dicer, an RNase nucleosubstitute family endonuclease, recognizes the dsRNA and cleaves it into short double-stranded fragments known as small interfering RNAs, siRNAs. These siRNAs are typically 25 nucleotides long and have 2 nucleotide overhangs at each 3 ends, end. RISC loading. The siRNAs are then incorporated into a multi-protein RNA-induced silencing complex, RISC. Within RISC, one strand of the cRNA, the guide strand, is retained while the other strand, the passenger strand, is degraded. The guide strand serves as a template for recognizing complementary mRNA targets. mRNA recognition and cleavage. The RISC, guided by the siRNA, binds to complementary mRNA molecules. The argonaute protein AGO, a key component of RISC, cleaves the target mRNA at a position opposite to the center of the siRNA guide strand. This mRNA cleavage results in the silencing of the target gene by preventing its translation into protein. Translational repression and mRNA degradation. Besides cleavage, RNAi can lead to gene silencing through translational repression or destabilization of the target mRNA without cleavage. This mechanism is more commonly associated with microRNAs, miRNAs, another class of small RNAs that share the RNAi pathway machinery. Amplification by RDRP in some organisms. In plants, fungi, and some animals, an RNA-dependent RNA polymerase, RDRP, can amplify the RNAi response by using cleaved mRNA fragments as templates to produce more dsRNA. This newly synthesized dsRNA is then processed by Dicer, generating additional siRNAs and further amplifying the silencing effect. Systemic spread in plants and some invertebrates. In plants and certain invertebrates, the RNAi signal can spread from cell to cell and even systemically throughout the organism. This systemic RNAi is thought to provide a broad defense mechanism, particularly against viral infections. RNAi plays crucial roles in gene regulation, cellular defense mechanisms against viruses, and the control of transposable elements. It also serves as a powerful tool in functional genomics and biotechnology for silencing specific genes to study their function or to develop genetically modified organisms with desirable traits. The discovery of RNAi has not only advanced our understanding of gene regulation, but also opened up new avenues for therapeutic interventions targeting diseases caused by genetic and acquired conditions. Exploiting non-homologous end-joining, NHEJ. Non-homologous end-joining, NHEJ, is a pathway for repairing double-strand breaks, DSBs, in DNA, which are among the most severe forms of DNA damage. NHEJ is critical for genomic integrity, functioning across different phases of the cell cycle, especially in non-dividing cells where homologous recombination, HR, is less active. Here's an overview of the chemical and biochemical mechanisms by which NHEJ operates. Detection of DNA double-strand breaks. The initial step in NHEJ involves the detection of the DSB. The Cu70/Cu80 heterodimer plays a crucial role in this process by directly binding to the ends of the DSB. This binding is highly affinity-driven and rapid, serving to protect the DNA ends from degradation and to recruit additional factors necessary for repair. Recruitment of DNA PKCs and formation of the DNA PK complex. Upon binding to the DNA ends, the Cu70/Cu80 complex recruits the DNA-dependent protein kinase catalytic subunit, DNA PKCs. The assembly of the Cu heterodimer and DNA PKCs forms the DNA PK complex, which is essential for the NHEJ process. DNA PKCs plays a role in aligning the DNA ends and preparing them for ligation. Processing of DNA ends. Before ligation can occur, the DNA ends may require processing to remove damaged or incompatible termini. This can include filling in gaps by polymerases such as PolMu and PolLambda, or removing nucleotides by nucleases such as Artemis. The goal is to create blunt ends or ends that are compatible for direct ligation. Ligation of DNA ends. The final step in the NHEJ pathway is the ligation of the DNA ends, mediated by the ligase to 4/XRCC 4/XLF complex. Ligase IV is specifically dedicated to the ligation step in NHEJ and works in concert with XRCC4 and XLF, Cernunos, to align and join the DNA ends efficiently. The XRCC4 protein acts as a stabilizing and stimulating factor for ligase I4, while XLF contributes to the ligation efficiency and is involved in the alignment of the DNA ends. Regulation and flexibility. NHEJ is characterized by its flexibility in handling a wide variety of DNA end structures. This flexibility is partly due to the various enzymes involved in the end processing step, which can adapt to different types of damage. Additionally, the pathway is tightly regulated by post-translational modifications, such as phosphorylation, of the involved proteins, which can influence their activities and interactions. Error-prone nature. Unlike homologous recombination, NHEJ does not require a homologous template and can be considered error-prone, leading to insertions, deletions, or mutations at the repair site. This aspect of NHEJ is particularly relevant in the context of genomic integrity and the potential for mutagenesis. Role in VDJ recombination. Beyond DNA repair, NHEJ plays a critical role in the immune system, particularly in VDJ recombination, the process that generates diversity in antibody and T-cell receptor genes. This process intentionally introduces DSBs, which are then repaired by NHEJ to create diverse antigen receptor repertoires. NHEJ is a fundamental DNA repair mechanism that allows cells to maintain genomic stability by efficiently repairing DSBs. Its ability to operate without the need for a homologous template makes it indispensable, especially in non-dividing cells, and for the repair of DSBs that occur during replication. In summary, each of these technologies offers unique advantages and challenges, and the choice of which to use depends on the specific requirements of the research or application in question. While CRISPR remains the most popular due to its simplicity and versatility, ongoing research and development in these alternatives promise to expand the toolkit available for genetic manipulation. Stay curious, stay innovative. I'm Luke McLaughlin. If you would like to learn more about this topic, consider reading our full-length articles on biotechnologyreviews.com, or consider our podcast series on YouTube or Spotify at our Biotechnology Reviews channel. Our articles are also available on our LinkedIn newsletter, Biotechnology Reviews.